I did my PhD here at Cornell in Dr. Claudia Fischbach's lab. So our lab is focused on studying how obesity, which is a major risk for breast cancer, and how that can actually contribute to the breast cancer progression. So that is kind of like my PhD work. So right now I'm kind of doing a 1 year postdoc, meaning research after PhD. So I'm kind of continuing on the same topic, but I just look at a slightly different angle. I guess I didn't really intend to go into cancer research because I was kind of intimidated in a sense, because I know it's so complicated. There are just so many puzzles. But I think eventually I'm still here, I guess. Eventually become a cancer researcher, and I think it is on one side is very scary, but on the other side it's also a fascinating science topic. So I think I'm still very fortunate to continue my study here, and hopefully eventually, maybe in 30 years, some of my PhD work may be translated into something that is useful for patients. So we'll see.
Very good. So I've made you a co-host, so hopefully you can share your slides.
Oh yeah, let me see. I think I yes.
Yes.
Okay. So I don't have too long, so about I think about 30, 40 minutes. So
Mhm.
not 1 hour long. So we'll see how that goes. So today I will be talking about the Barbara McClintock. She is one of the, I guess, when people talk about her, they always say she's one of the top geneticists here in the United States and also in the whole world. Basically, her research in the past, in the early 20th century to the mid 20th century, shaped our understanding about genetics. And she did all her work in the corn, and this is called the jumping genes that won her a Nobel Prize. And I'll talk about that later as well. So she was born in Connecticut in 1902. Her father was an army doctor, and her mother was a pianist. So you can see that she has three other siblings, and they all live happily together. So she actually moved to New York, well, Brooklyn, New York, when she was relatively young, and she attended all her schools here as well. So when she was still when she was young, she was very actually interested in science. But then when she decided to apply for college, her mother, according to the bibliography, was very concerned that she wouldn't find a husband if she go to college. I guess that's what happens in that time of the history. But then her father was supportive of her decision. So eventually she come here at Cornell and did her bachelor degree, Master of Science, and also a PhD. So she started in the, I guess, she did all her degrees in agriculture and botany. And then when she did her PhD, it's more focused on genetics. So at that time, she was the only female student in the department because they don't really admit people like they only admit male students because in that department you have to work with, I guess, farmers at that time, and they don't like to talk to female students. So it was kind of like she was one of the first to actually do that. And Dr. Lee Cass actually has a YouTube video online that you can check out as well. She is a professor at Cornell, and she kind of talk about like um, Barbara McClintock's life at Cornell in a very detailed way. So if you're interested, feel free to check that out. So I guess before we dive into all the things that she has done, I feel we need to have a little bit understanding about DNA and chromosome. That is the things that she has been worked on. So here is a really nice video. Sorry. Uh, about what this is, and I think we can just watch for about 1 minute here. Let me know if the sound works.
Stated clearly presents what is a chromosome?
So we can jump in
It's
here.
now time to talk about chromosomes. A chromosome is an entire chain of DNA along with a group of stabilizing proteins. You may have seen images showing DNA trapped inside a chromosome, sort of like this. This is not how chromosomes hold their DNA. If you were to unravel a chromosome, you would see that it's made of a thread, sort of like a spool of yarn. Zoom in again and you'll see that that fiber is a collection of protein orbs called histones, which are wrapped with a string-like structure. Zoom in on that string and you'll find that this is the chain of DNA. It is extremely long, millions of nucleotides long, and often contains hundreds of genes along its length. In this image, we're looking at a chromosome in its condensed yarn-like form, but chromosomes are only packaged tightly like this when a cell is reproducing. This structure helps with durability as the cell is splitting in two. For most of a cell's life, however, chromosomes exist in a loose, noodle-like structure along with other chromosomes safe inside the nucleus or centerpiece of a cell. Here is a photograph of an entire set of human chromosomes taken from a cell right as it was getting ready to reproduce. Your genome, the entire collection of genes that help make you who you are written across 23 pairs of chromosomes. That's 46 altogether. With very few exceptions, each cell in your body contains its own full copy of your chromosomes, your entire genetic code. Eyeball cells only use the eyeball cell genes, the rest are turned off. Liver cells only use liver cell genes, the rest are turned off, and so on for each cell type. One member of each pair of your chromosomes came from your mother, the other came from your father. If you were to examine the two individuals of a pair, you'd find that for the most part, both contain the exact same genes in the exact same locations. If you were to zoom into the actual genetic code, however, you would find slight sequence variations between them.
So I guess that's a very brief introduction about your DNA and how that is packed into this structure we call chromosome. And all the genetic code that you have is onto these chromosome structures here. And that is what Barbara McClintock was studying when she was at Cornell. Oops. So kind of before the early 20th centuries, people kind of know that there are some genetic materials, but people don't know anything about DNA or how this information is transferred from your parents to the daughter and sons. So before we have any understanding of that, scientists actually, he's not actually a scientist, but Gregor Mendel was the Austrian priest. He has a little garden he really like, and he's been studying different plants in the garden. One thing he noticed is that when he has these pea plants, he can see that the seeds are of very different shape. They can be round, or they can be wrinkled, or they can be yellow, or they can be green. And then the flowers can also be lower, or they can be higher. And it's not entirely random. So parent plants, where if you breed them and then have your daughter plants, then you see that there's a pattern to which this like structures are encoded. So he actually has a law of Mendel's. That's actually our first theoretic kind of very thorough dissected genetic information to study the genetics. And here's a YouTube video if you're interested to check that out. So I guess we can also take a look, but I don't know if people are interested to watch so many YouTube videos. It's a little tiny now.
Scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous P, where both alleles are identical, and what we call a heterozygous P, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's P's, for example. Let's write the dominant yellow allele as an uppercase y, and the recessive green allele as a lowercase y. The uppercase y always overpowers its lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous P mom will give each P kid a yellow dominant allele, and the green homozygous P dad will give a green recessive allele, so all the P kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
So that's a small summary about what Mendel did. Back when he didn't know what DNA is, he doesn't know anything about genetics. He's just trying to observe what the plants look like, and then he identified the code for genetics. So that is in the 19th century. But then because of that, nobody understands what's happening, right? So his theory didn't really get recognized until the early 20th century. And that is also followed by another giant in the genetics field, Thomas Morgan. So Morgan was a professor at Columbia. So what he did, instead of looking at the pea plants, he started to look at the fruit flies. So all the fruit flies actually have red eyes, but in one variation, the fruit fly actually had white eyes. And then he was following Mendel's experiment, and he accidentally discovered, or not accidentally, he discovered that only the male will have this white eyes. So then he discovered that you can have chromosomes, but the chromosomes also have sex chromosomes. So there are some traits that are only expressed when you are female or male. So that he actually went down to win a Nobel Prize. So at that time and this is in the early 1920s. So that is the era where Barbara McClintock was studying at Cornell, and that's how she became interested in the genetics of the corn. Oops. So that is another video if you're interested to check out. So at that time, so RA, yes.
When did he get his Nobel Prize? Did they recognize him right away?
Yeah, it was very big. It was very big. I think when he had the study published, he had a Nobel Prize not too long after that. Yeah.
Just benchmarking here.
Yes. So I think because his department, genetic department, was so successful, I think based on Dr. Cass' report, she suspected that Cornell wants to build something that's competitive to his department. So then Professor Emerson actually built this plant genetics department at Cornell around the 1920s and 1930s. And here are all the professors that are at the initial phase of the plant genetics department. So in the center is Emerson, and I believe the one on the left is Randolph, who was Barbara McClintock's PhD advisor. But it turns out that she didn't actually finish her PhD with him because they have arguments about authorship. So then she finished a PhD with another PhD advisor. I think it's him. I can't remember. It's hard to identify people from those old photos. But then basically she was very independent at that time, and she insists that her credit must be recognized. So she basically switched labs in the middle of her PhD studies, and that was very difficult at that time of year. So if you have people been to the botanic gardens at Cornell?
Mhm.
So now it's you have this very nice building, the Welcome Center. So if you start from the dairy bar, see, I don't know if you can see my cursor. If you can start with a dairy bar, you go down the Juteful Road, and then you have to make a huge loop, and then you go to the Arboretum Road, and down the side you can see the McClintock shed. So that is a little house that just stand there by itself. It is still there. That has a little plaque saying this is her field lab. So basically, this whole area used to be the cornfield at Cornell. So that's where she did all her studies of corn. But I think right now it's been converted into a botanic garden. So you don't see corns anymore. But if you go down by the road, you can still see the little house. That's just really not obvious, but you can see the little plaque. That is where she did her work here. Yeah. So I kind of mentioned this earlier before we started the talk that Cornell has a huge corn or maize group, not in terms of number, but in terms of achievement. So they study all kinds of genetics based on the corn and also look at how they can make the corn to be like a better crops and everything. So you can see Brava is on the right side standing here. And then this is Emerson. He's the head of the department. And then you have the Marcus Rhodes, who later became a professor at the Cold Spring Harbor that actually recruited Barbara later. And she became a researcher there for the rest of her career. And Charles Birmingham is also another professor, and he has been keeping constant letters, conversations with Barbara, and that's how we found out a lot about what she is thinking many years later during the different challenging years she had when she was trying to pursue her academic career. And George Beadle actually is another Nobel Prize winner, and he's also a geneticist. So he actually won the genetics, the physiology Nobel Prize much earlier than Barbara, like almost 40 years earlier, even though they were doing research at a similar time of like the era. So I think it really speaks to how late Barbara's achievements was recognized in the science field. So she didn't really study corn when she was in undergrad or master degree, but then she really started to look at them when she was in PhD. So if you look at the picture on the right side, you see that the corn is a very colorful crop, right? You see there are yellow trees, there are some purple ones, and you have a mixture of yellow and purples. So what Baba really started was not just those full color kernels. She studied those spotty kernels. So you have your entirely purple ones, you have your entirely yellow ones, and then you have those spotty ones. And that is kind of unusual, and that is what she studies. So when she was at her PhD, she kind of developed a staining technique, which she later used to actually discover the jumping genes. But then this technique allowed her to actually look at chromosomes to visualize what is being changed in the genetics of the corn versus what happened to the corn. Like if the chromosome look like this, do the corn become purple, yellow, or spotty? So that is actually very revolutionary when back in her time. And then after her PhD, she stayed on as an instructor at Cornell, kind of teaching all the students in the maize or the corn group and do research together. And while she was a PhD student, she also published the first map of the maize chromosome. So that is actually like very a big thing there. And then while she was an instructor, she also published a landmark paper on called what we call the crossover chromosome. So as you remember, the chromosome, when they become passed from the parent cell to a daughter cells, they kind of just duplicate cells. They become double, and then they split into two halves. So then the daughter ones will have identical genetic information as the parent, but sometimes they can cross over. As you can see, the legs cross over. So then you have some additional information on the chromosomes when it's passed to the daughter cells. So then when that situation happens, the daughters no longer has the same genetic traits as the parents. And that's when mutations and other things could happen. So she discovered this all in the corn, but this is a universal mechanism even in humans and many other organisms. And that is how sometimes the cancer mutations could also happen. But back then it was all done in corn and people didn't realize the importance of that. So then she went on trying to continue her academic career following her time at Cornell, even though she was very well established as a corn researcher on the genetics field. She didn't really get a lot of opportunities. But she did won a very prestigious fellowship, the Guggenheim Fellowship, which allowed her to study at Cornell as well as travel to Germany. However, if you notice the time is in 1933, so that is right before the World War II. So she actually had to stop her fellowship and then come back to the States because back there, nobody's really doing science anymore because it's just war and everything is tension. So then she lingered for a little bit because she couldn't find a job. But then when she did her fellowship, her postdoc advisor actually invited her to become assistant professor at the University of Missouri in 1936. So then she actually finally found a job that she was quite happy about. So in University of Missouri, what she did is that people know that if you shine X-rays onto different like organisms like fruit flies or corns, you can induce mutations. And you can then study what the mutations do. So in there, she actually discovered something very important called the breakage, fusion, and bridge. So here's a YouTube video describing that.
Welcome to biologyexamsforyou.com. Today we are going to discuss about the topic breakage fusion bridge cycle.
I think we can jump in a little bit.
First of all, let us have a look into a normal chromosome. A normal chromosome consists of a centromere and a pair of telomeres. A centromere is a region where spindle fibers attach during cell division. Telomere is responsible for maintaining the integrity or individuality of a chromosome.
Can you turn the audio up just a little bit on your end?
Oh, sorry.
PFP cycle starts with a breakage of a telomeric region. This, as in this figure, you can see right here, the telomere breaks off. As a result, the chromatid, the end of this particular chromatid chromosome is exposed. And this chromosome replicates, forming two sister chromatids, and both the chromatids are having a free end without a telomere. And this is the first step, that is the telomeric breakage. Now we have two sister chromatids that is having free ends that is attached to a common centromere. As there is no telomere, these ends has a tendency to fuse together. And a fusion occurs as in the figure. Fusion of sister chromatids at the ends occurs. Now during cell division, as this is a picture that shows that particular chromosome that is aligned at the center, you can see the spindle fibers that is attached to the centromere of that particular fused chromatid. During cell division, sister chromatids are separated and moves towards the opposite poles, and centromeric breaks. You can see right here, and these they are moving towards the poles. Ultimately forming a bridge. You can see right here, bridge formation, as this region is still intact. There is a bridge formation from one end to the another. This is called as a bridge, chromosome bridge. What happens after that? As the force increases, this bridge breaks, and this breakage can occur at any point along this line, and the daughter cells will be getting the same thing. The daughter cells receives chromosomes without telomere, and here also the telomere is absent, so that during the next division, in the case of daughter cells, the same cycle continues.
Okay, so that is kind of like the description of the brick, fuse, and the bridge that she discovered in the corn. So what happens here has a lot of implications in cancer actually, because normally you have this like the tail end, the tails that has a little telomeres that prevents them from like adhere together, tuck together. So now you don't have the end. So then the chromosomes, when they trying to split into two, they couldn't split. They become a little noodle bridge. And now if the cells continue to divide, it can break at any point. And that is how cancer mutations happen because it doesn't break at the right point. And the trouble continues because you don't have the ends that stop them from like tucked together. So in every single daughter cells, you have the same problem. So this kind of arrows or mutations will propagate. So eventually you just have tons of cells that are just doing random things that are not supposed to do. So she discovered all that in a corn. So she kind of realized some corns will have this kind of breakage of their chromosomes that is show up, and then she can study why they would do that. So in a corn, it may just change in color, but in humans, this may mean changing cancers. So there's a lot of implications and also conserved mechanisms that are happening between corns and humans. So this is while she was doing as a assistant professor at the Missouri. But then she wasn't really happy there. According to her letters with the other graduate student that just show you, Charles Burnham, she expressed a lot of frustrations about her job. She was excluded from all the faculty meetings and she wasn't getting the funding she requested. So she basically ended up in her letter described this situation as an inadequate opportunity for advancement. So Missouri actually ended up not giving her, they decided not to promote her to associate professor despite all her discoveries and scientific achievement. So then in 1940, she kind of took a sabbatical, meaning she's not doing research, but just spend the time trying to look up for something else. That's when she decided that she's not going to stay here any longer. She's going to find a new job that actually will make her feel happy at work and trying to actually allow her to do science. So that is when the other professor at Cold Spring Harbor, who was together with her at the maize group at Cornell, invited her to become a researcher rather than a professor, a researcher at Cold Spring Harbor. So she decided to take this offer and then she left Missouri. So then she moved to Cold Spring Harbor. Back then it was called the Carnegie Institute of Washington State Experimental Revolution. So actually within the same year, the station director decided that she was so excellent that she was appointed to a full-time position. So at that time, she was only giving a 1-year position, but then she got a full-time position, and then she spent the rest of her scientific career at Cold Spring Harbor. And that is where she discovered the jumping genes, which later is known as the transposons. So this she actually stayed in Cold Spring Harbor even after her retirement. So she has done research there for over 30 years. And then those are all the students and scientists and staffs over Cold Spring Harbor back in 1984. So she passed away in 1992. So she was still very active in science in her last few years of life. So what is a jumping gene? So we've been talking about the jumping gene. So jumping genes basically means the genes can jump between different locations of chromosomes. So the more scientific name is transposon or transposable element. So kind of just by judging the name, you can see that those are the elements that can move around within the genomes. So Barbara described them as the controlling element because she realized that those transposons or jumping gene can decide which genes is turned on or turned off. So that is how she described them. So what is the transposons in the corn? So here it may be a little complicated, but let me know if you have questions. So here on the right side, we have the pigmented kernel. You have the purple traits, which is what we call the wild type or the natural form. And sometimes you can come in and have a DS, which is the green part. That is one transposable element or transposon jumping gene. So if you have this jumping gene jump in and insert into where the purple genes are, then you get this yellow gene here. So now the kernels become all yellow. And then sometimes you have another jumping genes called the AC. So in the presence of AC, the green jumping genes can sometimes go out, sometimes come back, and sometimes go out and sometimes go back. And then now you have not just fully purple or fully yellow kernel, you can have this spotty kernel. And those AC jumping genes are very special because they themselves can jump in and out. So then if you just have those AC jumping genes here, then they can go in and out. And then your expression of kernel can be those spotty kernels as well. So she described those two types of jumping gene as a dissociator or an activator. So she presented her work at Cold Spring Harbor in 1951. So according to the counts at that conference, it was silence. People don't understand what's happening here. It was beyond imagination because back then people believe gene is constant. You have this gene from your parent, then your daughter gene is gonna be the same. But then she was arguing that the genes can free travel. They can jump around in different parts and then make the gene expression look very different. So at that time, it was very revolutionary. People don't were even arguing that her control, which is like the negative samples, were even wrong because they don't understand why would they be doing those things. So he wasn't really acknowledged about her discovery. So she actually stopped publishing things on this after 1953, and then she moved on to study other parts of corn genetics. And then it was not until many years later that the same phenomena was observed or discovered in fruit fly or bacteria and in the humans. And it was in the span of 30 years where scientists in other field of genetics field start to recognize that the genes can indeed jump between spots. And that is the jumping gene concept. And that's not until the 1970s that the concept of jumping genes is become then more accepted. So that is a very long time between the science discovery and the science recognition in the field. So the jumping genes, like I said, is very prevalent actually. So here is the mulling, I think it's called mulling glories. I forgot the name of the flower, but you can see that you can have those pure color flowers, but you can also have those stripes of different stripes. And those are all due to the jumping genes within the flower. And on the right side, you can see that in the fish, similar things could also happen. Like if you look at the fish eyes, they used to be just red, but because of the jumping genes, they become different colors, as you can see from here. So in those animals or plants, the most obvious changes for jumping genes are usually the colors, but they have many other implications. So jumping genes traditionally has been really implied in evolution because that's kind of how, if you have some kind of stress environment, it's the random insertion of those jumping genes maybe make you possess some special capacities or abilities so you can survive in those extreme environment, and those genes will actually be passed on to the next generations. So jumping genes is really important for evolution. And what's really striking is that 45% of human genomes is actually composed of the transposable elements or transposons or the jumping genes. But don't worry, most of them are inactive. So they kind of just sit there in your genomes and they don't do much. It was estimated that only 2 or 3% of them actually have some behaviors or some changes that may actually affect you. but those are the changes that really mattered in different diseases. So I think most of the jumping gene literature in cancer is focused, I think, in colorectal cancer. So like, for example, in this cancer type, which is a hereditary, meaning that those are like kind of genetic colorectal cancer types. Actually, more than 10% of them have researchers have found out that they have this kind of very weird transposon insertion. And then in renal and also smooth muscle tumors, you also have other types of insertion that are happening. So scientists actually make very funny names called Sleepy Beauty or piggyback. So those are kind of mechanisms of transposon or jumping genes that are happening in the different cancer mutations. So people discovered that those could happen in cancer because imagine you have your normal genes and then suddenly you have a jumping gene that's just jumping. And then you could have very different proteins that are produced, or you can have very different kind of mutations that are not supposed to be there. So then what scientists realized that we can actually back kind of reverse engineer the jumping gene mechanisms and use that to do cancer screening. So actually in colorectal cancers, people have used the sleeping beauty jumping gene mechanism to dissect it out at what stage, if you have this particular mutation, would you be have the cancer progression. So that is actually how people map out the four different distinctive genetic mutations in colorectal cancers. And that's actually one of the most well characterized like genetic mutations based on stages. So I think even though transposons most of the time they don't do much, when they sometimes do things in humans, it could be very disastrous. But we can also take advantage of this their properties to actually help us to understand cancer better. So that is kind of like the implications of the jumping genes in cancer. So as I mentioned that scientists, it took us other scientists more than 30 years to catch up what Barbara discovered in the 1950s. So she actually, after she has been receiving a lot of awards just based on her achievements in the cornfield. But then it was not until the 1970s then she became recognized on her contributions to the genetics field in general. And the jumping gene actually led her to become the first woman to be awarded the National Medal of Science. And subsequently in 1983, she was awarded a Nobel Prize for her discovery of this mobile genetic element. So I think there's also a YouTube video about her Nobel Prize speech online. So I think this image was really nice. I was really interested in that. So this was taken in 1989, shortly after she won the Nobel Prize. So she was taken with two other women. So Dr. Fedorov on the right side, so Barbara hypothesized that this is the transposon or the jumping gene. So Dr. Fedorov was actually the first one to actually understand what is the molecular mechanism behind it. And then uh, Dr. Maxine Singer was the first one to actually identify that in the human genome. So all the scientists come together after they realize what's really happening, and then they can keep expanding our understanding of the genetic field. And they're all female scientists, so I thought that was pretty cool. Um, and of course, after she won the Nobel Prize, she became very famous. So reporters keep chasing her down, and then she has to do tons of different interviews, which, according to her letters, she doesn't really like. She preferred to do science by herself in her little lab, but then she is famous now, so can't escape the fame afterwards. So, but I think what's really interesting is that she continued her research when she was really discouraged in the 1950s. She then became interested in the corn development. So she actually has a very strong bonding with researchers in the Latin America, in which she kind of often go to Mexico to trace the origin of the corn. and then to try to understand how can they engineer the corn to actually make the corn grow better from understanding their genetic materials. And then Professor Robert Mattison was actually I think, one of her students at Cold Spring Harbor, and now he's also a professor. And he has this quote, I think that was really nice. So he described her as someone who's really fierce and very hardworking doing science work, but at the same time, she was very generous in mentoring the different students, and especially women in science, to kind of expand their and kind of pave the way to allow them to kind of continue in the science without having to, I guess, trying to encounter so many obstacles like what she did during her scientific career. And I think in terms of legacy at Cornell, so right now we have the McClintock Graduate Student Award for all the senior graduate students who study the genetics here. And then we also have the life science lectures, which actually we have one next tomorrow. So it is established to kind of celebrate the most distinguished scientists, and they only invite 4 scientists per year. And then they kind of invite them to kind of discuss science in a very multidisciplinary way, or kind of to combine the different natural sciences, physical sciences, and everything. So I saw this initiative to establish the life science lectures was also really nice, considering her contribution to the science field. So I guess to end this, I have 2 quotes from her that I really like. So Barbara was talking about first what happens if people don't believe you. So she said, over the many years, I truly enjoyed not being required to defend my interpretations. And she would work with the greatest pleasure, and she doesn't feel she need to defend her views. If she's wrong, then she just forgot she has this. So I will say that not all her scientific views are correct, but the ones that are actually correct are correct. So I think this is a really nice quote. And then the last one is that she just really celebrated her science. She said, I have been so interested in what I was doing, and it has been such a pleasure that I never thought of stopping. I had a very, very satisfying and interesting life. So I guess I'll end here with the little life stories about Barbara McClintock.